Hey everybody, my name is Brian Markowski, and today I'm going to be talking about long-lived proteins and memories that last a lifetime. Memories are formed by constant chemical and electrical signaling within the brain at synapses. Many different types of proteins are found at these synapses, and they are believed to play an important role in memory. The question addressed by Johns Hopkins University in this study was, how can some memories last a lifetime if most proteins rapidly die and renew at these synapses that we believe are important for memory? Johns Hopkins University studied 8 mice extensively over the course of 7 weeks. The researchers put a molecular tracer in the mice's food, and the amount of molecular tracer was either heavy or light. Mass spectrometry was used to analyze the proteins that had higher amounts of molecular tracer throughout the 7 weeks. They discovered 164 proteins within synapses that lasted weeks to months longer than the average proteins. Some of the mice were forced to stay in their cage the entire time, while other mice were allowed to explore an enriched environment in order to promote neural activity. Researchers expected to see protein stability in the mice exposed to the enriched environment due to increased neural activity. However, they collected evidence that suggested that exposure to the enriched environment actually led to increased protein cycling or decreased protein stability. Also, they inhibited neural activity, which resulted in increased protein stability in a separate experiment. The future goal of this research is to follow about 50 of the longer-lasting proteins that they discovered and make connections to age-related cognitive decline in humans. One thing that really stands out about the primary research article is that it is more specific and detailed compared to the published press article. The primary research article lists specific synaptic proteins and pathways that the general public would most likely not understand. The website is neurosciencenews.com, so the audience probably had at least some background in neuroscience, but I still think it is a good move to focus on the broader points. I think the news article could have explained the purpose of the 2 different environments the mice were kept in a little more clearly. The news article also failed to mention the results based on their living environments, which I thought may have been important enough to conclude in the article. I liked how the news articles brought in the figures from the primary research article to help the audience visualize their findings. Overall, I thought the research summarized by this article could be very beneficial in future research regarding memory.